Chapter 9. Oncogenes. This chapter focuses on genes that, when mutated or overactive, contribute to cancer by driving uncontrolled cell growth. Oncogenes originate from proto-oncogenes, which normally regulate cell division. When these genes become dysregulated, they promote survival, proliferation, and tumor formation. Understanding oncogenes is key to diagnosing and targeting cancer more effectively. Proto-oncogenes are essential for normal cell growth, signaling, and division. These genes are tightly regulated in healthy cells. However, when mutated or overexpressed, proto-oncogenes become oncogenes, genes that promote cancer. Oncogenes can drive excessive proliferation or prevent normal cell death. This transformation is a critical step in the development of many tumors. There are four main mechanisms through which proto-oncogenes become oncogenes. These include point mutations, gene amplification, chromosomal translocation, and insertional mutagenesis. Each mechanism alters the gene's behavior, either by boosting expression or modifying protein function. These genetic changes give cells growth advantages that support tumor formation. The activation of oncogenes marks a major shift toward malignancy. A point mutation is a single base change in DNA that can dramatically alter protein function. One common example is a mutation in the RAS gene, which results in a protein that is permanently switched on. This leads to continuous stimulation of cell division pathways. Even one such mutation can significantly disrupt normal cellular regulation. Point mutations are among the most frequent oncogenic changes seen in human cancers. Gene amplification refers to the creation of multiple copies of a proto-oncogene. This results in overproduction of the encoded protein, which drives uncontrolled cell signaling and division. A prime example is HER2/NOI in breast cancer, which is overexpressed in a subset of patients. These patients may be treated with trastuzumab or Herceptin, a monoclonal antibody that targets the HER2 receptor. Targeted therapies like this rely on understanding the mechanism of gene amplification. Chromosomal translocations involve rearrangements that place oncogenes under the control of new, more active promoters. A classic example is the Philadelphia chromosome, a fusion between BCR and ABL genes seen in chronic myeloid leukemia. This fusion results in a continuously active tyrosine kinase enzyme. Such translocations are highly specific to certain cancers and can serve as diagnostic markers. They also offer precise therapeutic targets. Insertional mutagenesis occurs when viral DNA integrates into the host genome near a proto-oncogene. This can disrupt normal gene regulation, leading to overexpression of growth-promoting proteins. It is often seen in retroviral infections and serves as a model for understanding how viruses can initiate cancer. The random nature of viral insertion makes this a dangerous but informative process. Insertional mutagenesis underscores the link between infection and genetic instability. Several oncogenes are frequently involved in human cancers. RAS mutations are found in roughly 30% of all tumors and influence cell signaling. MYC is a transcription factor that drives cell proliferation. HER2 is involved in breast cancer, and BCR-ABL is specific to leukemia. These oncogenes illustrate how diverse mechanisms can converge to support cancer development. Most oncogenes function within major signaling pathways that regulate survival, proliferation, and angiogenesis. When activated, they can bypass feedback loops and sustain growth signals indefinitely. This leads to a cellular environment that favors tumor progression. These pathways are complex and interconnected, making them both powerful and difficult to regulate. Understanding these routes is essential for designing targeted treatments. Unlike tumor suppressor genes, which usually require both alleles to be inactivated, oncogenes often have a dominant effect. A single mutated or overexpressed copy is sufficient to drive cancer-related changes. This makes oncogene activation a potent force in tumorigenesis. It also explains why many targeted therapies focus on blocking the action of a single hyperactive protein. This dominant effect is what makes oncogenes both dangerous and therapeutically relevant. Some cancers become highly dependent on a single oncogene for survival and growth, a concept known as oncogene addiction. This creates an opportunity for targeted therapy where blocking that one gene can cause cancer cell death. An example is the use of imatinib or Gleevec to target BCR-ABL in leukemia. When this oncogene is inhibited, the cancer cells die off rapidly. This selective vulnerability has revolutionized treatment for certain cancers. Therapies targeting oncogenes include monoclonal antibodies, tyrosine kinase inhibitors, and emerging technologies like CRISPR. For example, trastuzumab targets HER2 positive breast cancer, and imatinib targets the BCR-ABL fusion protein. RNA-based therapies are being developed to silence oncogene expression at the transcript level. As we understand more about oncogenes, our toolkit for treating cancer grows. Personalized medicine depends on matching the right drug to the right genetic target. Despite early successes, targeting oncogenes isn't without challenges. Cancers can develop resistance through secondary mutations or activation of alternate pathways. Signaling networks are highly redundant, meaning that blocking one node may not be enough. Combination therapies are often needed to prevent resistance and improve outcomes. Continued research is vital to overcome these limitations. Oncogenes also serve as important biomarkers in cancer diagnosis and prognosis. Testing for specific gene mutations or amplifications helps guide treatment selection. HER2 status in breast cancer, for example, determines eligibility for trastuzumab therapy. These genetic markers enhance precision in modern oncology. Incorporating molecular diagnostics into routine care improves patient outcomes. Oncogenes are central drivers of cancer progression. Their activation leads to unregulated growth, survival, and invasiveness. By understanding their mechanisms, scientists have developed highly effective therapies for specific cancers. Continued research into oncogene pathways holds the key to even more targeted and effective treatments. In the era of precision medicine, knowledge of oncogenes is essential.